Welcome, Omics Group. I would like first to express my appreciation and thankful to Mr. John Winston, who helped me so much and facilitates all the paperwork enabled to attend. But unfortunately, for several reasons, I couldn't come. Also, please allow me to say thank you to all of you in giving me this times an opportunity to talk about the coronavirus and the cases in Saudi Arabia. And I would be so grateful to you if you pay attention for 10 to 15 minutes to my lectures, which may add something to you. And I would like to come certainly and have a discussions, because there is so many points needs to be discussed. I know this virus, there is so many controversial issues around it, but I couldn't make it because, as I said, there is so many difficulties and several issues come to the surface regarding my coming. The visa, my illness, which I get bronchitis, last 3 days before. I'm going to start first showing a brief video, just to motivate you to follow me in this lecture. Okay, I will start now my lectures by giving the aim of my study is to raise the point of view on MERS or coronavirus. So the statistical analysis for all my project was based on the statistical analysis. So the aim is to raise the point of view on MERS-CoV statistical analysis and evaluate how the virus can firstly cause the infections. I forgot to give an introduction of myself. My name is Ilham Qattan, Assistant Professor of Medical and Molecular Virology, Dean of Medical Applied Science, and Deputy of the University General Supervisor for Public Relations and Conferences and Media, Board Member of the Genes and the Genetic Disease Centers at the Taiba University in Medina Munawwara, Kingdom of Saudi Arabia. Okay, I will start first with a brief introductions about the coronavirus or the MERS virus. In September 2012, coronavirus was identified for the first time in a new series of infection known as a Middle East respiratory syndromes. Is considering a new complicated disease infected the epithelial cells in the respiratory and intestinal tract, and thus causing a disease in epidemic proportions. The incubation period is either a short between 2 to 4 or 7 days, or between 12 to 14 days. Okay, when I wanted to classify, or the classifications of this virus, the virus is a species that belongs to a subfamily called Coronaviridae. The virus is an envelope with a single strand, positive sense RNA. The genome has a helical symmetry of their nucleosides or nucleocapsids. The genome size range from 26 to 32 kilobase. It is one of the largest RNA viruses. A brief history: since March 2012 to the end of November 2015, around 1,655 cases were infected. 577 were reported deaths, and 630 were recovering. Some of the cases, in around 28%, were having non-applicable data. This is, of course, reported by WHO. The virus has been detected in Arabian Peninsula, European countries such as Britain, Germany, France, Italy, and Middle East. As you see here, the first case was reported in 21st of March 2012 for a 40 years female nurse working in Jordan. Then in the 5th of October 2012, the second case was reported in Saudi Arabia in Al-Ahsa for two family members. Then the cases were subsequently distributed between Tunisia, Italy, France, United Kingdom, United Arab, Oman, Netherlands, Iran, Korea, United States, Lebanon, Egypt, and China. Factors relating to the patients and variation of the virus through mutations and integration with other species might play a role, of course, in creating more malignant infection, which can lead to death. This study is based upon the global statistical analysis published online with a special focus in Saudi Arabia. All cases were compared to examine the differences between infection, recovering, and death in order to determine the significant factors that could be explained how the infection spread. Because of the lack of the infection, because of the lack of the source of information, the statistical reports have been monitored for data collection starting since the beginning of the infections and have been updated frequently. The data were collected through Ministry of Health, the Command and Control Center, World Health Organization, Center for Disease Control, the CDC, and European Centers for Disease, in addition to a social networking such as Twitter, Facebook, from all over the world, and also collecting the data from the publishing articles and news. As we know, the genes of this virus were compared with the genes similar to the viruses from human, pigs, cows, dogs, cats, mice, rats, camels, and pets. It was clear that each gene has more or less greater than or equal 70% identical compared to the other gene with the same family. Data from studies indicated that the new mutated virus was dormant in animals and coexisted with them for a long time. Then somehow it was isolated genetically for a long time, and then suddenly become a malignant virus reported and harmful towards humans. The transmutations to humans might occur only at once, and it could be due to the dodgy mixtures between species and random mutations. Therefore, it then can affect humans and original host. The phyloanalysis tree shows that the theory comes up to the surface regarding the transmissions of the virus. The phyloanalysis tree of human-associated coronavirus show are related to other known variations of corona or toroviruses. These studies began by Percy et al. In 2003. His results shows that corona can undergo transmission, and further analysis were shown that the virus with coronavirus can change when they come into contact with different family groups. This is very important to understand. The genetic analysis shows that this can happen also within the genomes, and the combination of any genome or any genome changes leads to a new kind of coronavirus. And the genetic or clinical data, therefore, shows that the original genes can be no longer recognizable. I'm so sorry if I couldn't raise my voice because I have very severe bronchitis. And I'm trying to fulfill my honesty to Mr. John, who helped me a lot. And I would like just to give lectures and to show my studies, which is very important to me to present it in this omics. Okay. The MERS hits 27 countries during from 2012 to 2015. Those countries from Germany, Iran, Spain, from all over the world. The total cases shows that 1,655 cases all around the world, and the most cases were in Saudi Arabia, shows 1,308. The total of deaths is 481, and the recovery were reported with 470 cases. Some of the countries were having a report a case, but they are not following the case if it is death or recovering. So there is some gaps or data non-applicable because of the reporting from different countries, or sometimes the WHO having non-complete report due to the Ministry of Health of each country that they are having insufficient reporting of the cases. Since March 2012 till the end of November 2015, as I said earlier, 1,655 cases were reported, 87% in the Middle East, and 1,039 cases were reported in Saudi Arabia. Of 91 cases, 91% the percentage of the cases infected in Saudi Arabia. The number of the deaths were 557 of 340% from all over the world were reported, and 514 of 92% of the cases were reported from the Middle East. Most of the death cases were from Saudi Arabia. Of the total 481 cases, and 86%. The number of the recovering were 630 cases, representing 38%, and 481 cases of 76% were in the Middle East. And this graph shows that the number of cases and deaths, the green column shows recovery cases, and the red columns shows the dead cases, where the blue columns in the each row shows the asymptomatic cases. Unclassified cases shows in the gray columns between 2012, 2013, 14, and 15. This is the number of cases were reported. The cases were correlated with the seasons during springs. It shows here that the spring season was finding that it is significantly correlated with this spreading of the MERS corona, and the majority of the cases were 49%. Approximately half of the cases were distributed during spring. Whereas spreading in winter and autumn were 12%, and in summer around 26%. In 2012, the spreading of the cases was during autumn, and high peak was in the October, where in 2013 the spreading was during summer and autumn, and the high peak was in September. In 2014, the infection was too high during April, and in 2015 the spreading was during February, January. It is dropped down in July and become high again during August. I think it is in this graph. I will repeat it again. In 2012, the cases were high in October, autumn in Saudi Arabia, and in 2013, the highest cases was reported in September, where in 2014, most of the cases were distributed between April and May, and the high peaks shows in April. In 2015, some of the cases were reported in February, January, and goes down in July, and again the cases were reported high during August. The distribution of MERS corona within the Kingdom of Saudi Arabia region shows that Riyadh had an infected cases of 617 cases, and the deaths were 208. Recovery cases were 221. Mecca region shows 361 cases. The deaths reported with 104 cases, and the recovery cases reported as 64. When we talk about Medina region, 61 cases were infected, 19 cases were reported death, and 15 cases were reported as recovered. This graph shows the distributions between male and females with the age. And of course, all the reports, and this also in the statistical graph shows that the males are more likely to get the infection more than the females, of 66%, and the highest number of the infected cases were between age 50 to 59 years old. This graph shows the accumulations of the MERS coronavirus cases, and this in all over the world between 2012 and 2015. And this case, this graph shows the accumulations of the MERS cases and deaths among Saudi Arabian populations, also between 2012, 13, 14, and till the end of November 2015. So many issues regarding the transmissions. Researchers till now couldn't find the right route of transmitting this virus. The cases in Saudi Arabia were distributed so many very fast, and the deaths also regarding several issues. Probably by contacting to animals such as a camel, or by health worker, or by direct contact. From all the statistical data, we find that 13% of the health course, 13% were transmitted, the virus were transmitted via health worker. And the virus were transmitted through direct contact in 17% of the cases, whereas by air in 18% of all around, all over the world. In Saudi Arabia, it shows that only 5% of the cases had direct contact with camels, whereas 8% of all cases had contact with different types of animals like sheep or birds or dogs. But till now, there is no accurate research investigating this phenomena or investigating this this percentage of the cases. Most of the research is mentioning that the camels is the main source of transmissions. This could be right and this could be wrong. It depends on the cases and it depends on the research and investigations. The research based on the camel's transmissions is due to the antibodies isolations in different studies, but not sure if they are isolated the virus rather than the antibodies. Number of the direct contact to patients with families was 258 cases, where from air was 268, and from health worker was 240. The statistical studies have shown that the cases of the infections and the deaths within the geographical regions were associated with the mode of transmissions, and these were able to count the number of the confirming cases and deaths worldwide. The analysis also shows that the virus attach to a seasons and affect the age and gender on the infections, like male more than females. And the infections found that the more males are more susceptible to the infections because they are going out and having some working rather than the women here in Saudi Arabia. They're staying at home and the going out is so very limited to them. Also, the statistical analysis shows that the infection increase among the earlier old ages and those having the greater than 40 years of age, and more than due to the infection. The infection were distributed among them because they were having some chronic diseases like kidney failure or heart failure or some TB chronic infections or past exposure to any of the respiratory tract infections. The data collected from all over the world and available online. But what I did here in this study, that I pull all the data together and filtered it according to the reported cases from Ministry of Health and the WHO and the CDC. Only these reported of the cases are available. Samples are not available and prohibited to go. It was prohibited to get any researches during the outbreak, but now I think it is available to have some research according to the Ministry of Health rules and regulations. They allow now some of the group to have some researches on the coronaviruses to put some clarifications how this virus is transmitted and why this virus is transmitted in such in such a way that it has come suddenly and disappear suddenly. The data collected was, as I said, from the online source and provide only a limited statistical information, which cannot also explain how the virus is transmitted. The only way that we know is the preventions by following the precautions according to the CDC and the infections control procedures. Of course, if we're talking about the research, it is still in progressing, and the data and the sampling for the disease has been limited, and some were non-applicable until now due to the misgapping of the reports. If we're talking about the research, the ancestral sequencing of the MERS genome and the host polymorphism association analysis might show the presence of the humans and the camel, the presence of the virus in human and the camels, and might show which allele is responsible for distributing and transmitting the virus. Also, hierarchical clustering analysis in associations with heterogeneity or topology tree can introduce a model to support the evaluations of the mode of transmissions of this virus. Some of the reported, like epidemiological reported or case reported, can help also to give an idea how many cases are infected and how many cases are not infected. As I said before, the misgapping of reporting cases and the misgapping of getting into the samples and the misgapping in contacting to right source of medical institute is making a lack of informations. What I'm trying to say here that if we are opening a field of gathering together with all the virologists from all over the world and having complete discussions how these viruses can be transmitted by pulling some samples and having protein expressions or having complete genome sequencing from different type of animals and humans, doing hierarchical clustering with the topology tree, comparing also polymorphism with the alleles coming from humans and animals, can give us a light or can highlighting how the virus can transmitted and why the virus transmitting during some seasons, and maybe can reaching to a good points of clearing or making clarifications of these viruses. The vaccines, of course, are requiring, and it's very important, but it is due to as I said, to the lack of getting samples and lack of research, is making it very difficult to understand how this virus is moving and how this virus is distributing. I would like to thank you all for listening and paying attention to my lecture. It's very helpful lectures, and I just wanted to say thank you to you all. Thank you, Mr. Jones, for giving me opportunity to present my data and my lectures. I would like to come by myself, but as I said, that due to my illness and several issues of getting visas and commitments to work, so I would ask you please to present this lectures to the group of omics, and if there is any discussions or any questions, please email me or send me or call me by Skype or use my website to contact me. I would be happy to discuss any questions or finding any solution, or also I would happy to become participants in any research group would like to have full investigations or some investigations on the MERS coronavirus. Thank you for paying attention, and see you soon in different conferences. Thank you.